As the cell goes through the processes of DNA replication and protein synthesis, sometimes mistakes happen, and those mistakes are called mutations. This is when we get a change in the DNA sequence of an organism that can be inherited. The resulting organism is called a mutant, and the original one is called the wild type. That's what we commonly observe in nature, but sometimes we get mutations. This process is not perfect, even though there are some correction enzymes and correction procedures in place to help clean up some of those mistakes that can be made. Maybe not all of them are caught. If we get a change in the amino acid sequence of a protein, it more than likely will affect the function of that protein. In the protein world, we say structure determines function, and if the structure is different than it should be, then that protein may not work like it should. And this can lead to an altered phenotype, or how that gene is going to be expressed. When we look at mutations, there are several different types. We have point mutations that affect a single base, and then we can have these frame shift mutations that can involve insertions or deletions. So if we look at some of these. So a point mutation is just a substitution of a single base, and the silent type of point mutation has no effect. Remember that we have some redundancy in that genetic code, and in this example here, we should have this A here, but we've got a mistake of having a T. And so when this is all translated, we get the wrong amino acid that goes in here. Okay, so we get the mRNA is wrong, or I'm sorry, not wrong amino acid, the nucleotide is wrong. And with the nucleotide being wrong, that could potentially affect the amino acid, but because of that redundancy, it doesn't. And so you can see that we have serine, valine, proline, and tyrosine, serine, valine, proline, and tyrosine. So we don't get any effect on the protein sequence. So the base is wrong, but it doesn't affect our amino acid. However, in this example, we get, instead of CCC, we get ACC. This is called a missense mutation, because now instead of proline here, we have threonine. So now our protein sequence is wrong. It is not what it should be. So that's called a missense mutation. A nonsense mutation is when we put in a stop codon where there should be an amino acid. So here we have a G instead of a C. So instead of UAC and tyrosine, we have UAG, which is a stop codon. So what's going to happen is the process is going to stop right there. So we get an incomplete protein. It could be correct up to that point, but it's going to be incomplete. It's not going to be finished because it got to that stop codon before it should have. In a frame shift mutation, it affects the reading because now we're going to insert or we're going to delete, and the reading frame is shifted. And so past the point of the insertion or deletion, we could potentially get a change in all of the rest of the amino acids. So what happens here, so here's our normal, but what happens is the TA is removed. It's left out. It's just completely skipped. So the serine is the same, but all other amino acids in the sequence are now going to be different because two of the bases have been removed. So if we insert or if we delete one or more bases, it shifts how the ribosome reads the code and puts in a lot of wrong amino acids. So again, this protein is not going to be able to function as it should. Now, sometimes we can get beneficial mutations. Not all mutations are bad. Sometimes something happens and an individual gets a mutation that turns out to be beneficial in its environment. That organism, survives and passes on that mutation to, future offsprings. So, what causes mutations? Well, sometimes we just get spontaneous mutations. You see there the error rate of DNA polymerase is 1 incorrect base per billion base pairs replicated. So just in normal replication, not being exposed to any type of chemical or physical mutagen, there's 1 incorrect base per billion pairs replicated. So overall, pretty good success rate at getting it right. However, if the cells are exposed to mutagens, this can induce mutations and can increase this rate of mutation more than 1,000-fold. So if we look at some of the types of mutations, chemicals can interfere with the process. Nucleoside analogs are chemicals that are very similar to normal nucleotide bases. And instead of the DNA polymerase picking up the correct nucleotide and putting it in, it picks up one of these analogs. And so they can induce mutations because of the base pairing rules. Some chemical mutagens can modify normal DNA bases, also resulting in some different base pairing rules. We also have chemical mutagens called intercalating agents, and they basically get in between the nitrogenous bases in DNA, which distorts it, and as it's going through its process of replication, there's some different spacing in there, and the DNA polymerase is either going to skip or skip replicating several nucleotides or insert some extra pieces in there. So here's a normal nitrogenous base. This is an adenine nucleoside here and an analog, so it's very similar. Okay, you see very similar here. And so what would happen is the DNA would pick up this analog and put it in place of that. And so what that's going to do is it's going to interfere with the correct process. So we can have these chemical mutagens, radiation. Ionizing or non-ionizing radiation can cause mutations by different mechanisms. Ionizing radiation is things like X-rays or gamma rays. Basically what happens is they can cause breaks in the DNA backbone. So that sugar phosphate backbone, they can cause breaks in that. Ionizing radiation can also modify bases, changing, for instance, cytosine to uracil. And we'll talk more about ionizing radiation in when we get to chapter 13, when we talk about methods of microbial control. Non-ionizing radiation, which is like UV light, induces what we call dimer formation. And this commonly happens between two thymines, so we call this a thymine dimer. Essentially what happens is those two thymines become linked. A dimer is when we have basically two of the same structure that are linked together. And so the thymines should not be connected to one another, but they're beside each other and they become linked together with a bond. And if that isn't fixed, DNA replication and transcription can't proceed past that point. And if the DNA polymerase continues, then that's going to be replicated incorrectly, and that leads to mutation. So radiation is another form of or a type of mutagen.